Our project is cAMP and calcium ion transport. cAMP, or cAMP, is cyclic adenosine monophosphate, which is a small molecule made from ATP. cAMP is a secondary messenger, and it activates an enzyme called protein kinase A, or pKa, which enables it to phosphorylate its targets and pass along the signal. cAMP is regulated by the activation of G protein-coupled receptors, or GCPRs, and it is inactivated by enzymes called phosphodiesterase. So the process of cAMP begins with the activation of a G protein, which releases an alpha subunit, and that alpha subunit activates adenylyl cyclase, which transforms ATP into cAMP. And cAMP then activates protein kinase A, or pKa, which phosphorylates its targets and passes the signal down the pathway. And cAMP is inactivated by enzymes called phosphodiesterase, which break the ring of cAMP and turn it into just AMP. So calcium ions are another type of secondary messenger. They're the most common secondary messenger. And calcium ions typically travel via a protein channel across the cellular membrane because the cytosol typically has a low concentration of calcium, so they flow down the concentration gradient through a protein channel. And calcium ions often bind to receptor sites, which causes the shape of protein to change, thus activating the protein. And many of these proteins work to contract muscles and perform procedures such as apoptosis and excitosis and more. And so the process for calcium ion transport begins with a ligand being released by upstream signaling events, and those ligands bind to ligand-gated calcium channels and open them. And high levels of calcium ions flow into the cytoplasm, increasing the lower concentration inside the cell. Proteins with calcium ion binding sites then allow calcium ions to bind to them, which changes their structure and function. And an example of calcium ion signaling is in the beta cells of the pancreas to release insulin.
Go.